We have seen the effects of treadmilling when we were talking about treadmilling in the previous module. Dynamic instability is also a classic character of our cytoskeletal filaments. It's just that dynamic instability is more associated with microtubules than it is with actin filaments. So let's talk about dynamic instability. If the concentration of free subunit is intermediate between critical concentration for D form and the critical concentration for D form, a microtubule end that happens to be in T form will grow, whereas any end that is in D form will shrink. On a single filament, an end might grow for certain length of time in a T form, but then suddenly change to D form and begin to shrink rapidly. That can happen, for example, if addition of monomers at that end slows down and the rate of hydrolysis of we are talking about microtubules, so GTP into GDP catches up. So your microtubule plus end will be in D form, and that will start to disassemble or depolymerize. At a later time, the same microtubule might then again regain the T form of the polymer, T form of the subunits, and again begin to grow again. So these microtubules, they keep on switching between the growth phase and the shortening phase. So these structures are not constantly either growing or shortening. They switch into different modes after a short while. Dynamic instability is basically the rapid inner conversion between growing and shrinking state at uniform subunit concentration. Don't worry, I will show you again a little video of this dynamic instability, so the concept will become more clear. But terms I do need to tell you about. Catastrophe is the change to rapid shrinkage. Rescue is change to growth. In cells, dynamic instability is predominant in microtubules, whereas treadmilling may predominate actin filaments, as I mentioned earlier. Here, for example, you have a diagram in which you have a GTP capped end of the microtubule. So those solid green squares at the end are basically the microtubule subunits. And this part of the microtubule is in T form, whereas this part is in D form. So we say that this microtubule has a T-capped, GTP-capped or T-capped microtubule. So accidentally, what happens is that for some reason, the subunits don't add on as quickly, or basically the rate of hydrolysis increases for some reason. So you end up losing that T-cap that you had, the subunits which were bound to GTP. And as a result of that, we have already seen the critical concentration for D form of the polymer is much higher, meaning that now the subunits will start, this filament will start to lose the subunits. It will lose subunits, and at certain point, the subunit loss will pause. That is basically the rescue part. You're losing the monomer from the subunits. These addition or loss, they're in equilibrium, just like a reaction. It can go both ways, forward or backward. At certain point, if there is a pause, you can add, if a new subunit is added, which is in T form, you suddenly will have a spurt of growth in the filament. So this is basically a diagram which shows dynamic instability in the graphic form. Things I'll be talking about, I thought I'll show you, but now we'll talk about some of the features of dynamic instability. Tubular subunits with GTP bound to beta monomer produce straight protofilaments that make strong and regular lateral contacts with one another. But hydrolysis of GTP to GDP causes a conformational change which makes the protofilament curved. T form, straight protofilament. D form, protofilament has a curvature which will tend to split subunits which are making lateral contacts with each other. I'll show you a photograph in the next slide. GTP cap of readily growing microtubule constrains the curvature of protofilaments at the end. The ends appear straight. But when the terminal subunits have hydrolyzed their nucleotides, this constraint is released and curved protofilaments spring apart. So here, let me show you this. So here you have alpha and beta subunits. The beta is bound to GTP. The beta, the subunits, when they polymerize, they are in T form and the protofilament is straight. These subunits have GTP as activity. When these subunits have hydrolyzed GTP into GDP, by default, there is a conformational change. You have lost a phosphate group, and this protofilament will become curved, and the lateral contacts this protofilament was making with adjacent subunits, they will become weak, or they will be lost, because the default curvature of GDP form of the filament. When this happens, the microtubule will depolymerize. Let me show you another photograph. So here you have the T cap, the GTP cap. This is keeping the microtubule end straight. You hydrolyze, these red dots are beta tubulin bound to GTP. If it hydrolyzes GTP, the protofilaments by default they become curved, because they are in a different conformation, and when that happens, this microtubule will start to shrink. It will start to depolymerize, because these bonds, the lateral bonds which are holding these monomers, these lateral bonds, they will become weakened because of the curvature, and the microtubule becomes less stable and starts to depolymerize. Not convinced? I'll show you an electron micrograph. Here is the electron micrograph, growing microtubule and the shrinking microtubule. You can see the curved protofilaments, which are hallmark for microtubule that was depolymerizing. Now I will like to show you an curved microtubules, which are hallmark for microtubule depolymerization. The curved protofilaments we saw in the EM. I would like to show you an animation of this dynamic instability, actual microtubules in cell going through bouts of assembly and also depolymerization. Let's watch that animation in the next module.